Okay, so this course is on Proteomics Informatics, and so we will have it several parts to it. One is that we'll always have a small lecture, maybe an hour, maybe less, and then after that there will be more a practical part that will involve programming, and so we'll start from the beginning to teach you how to program in Python. And does anyone know how to program anything? So good. And so you know, yeah. So you'll learn during the, and the idea is that since it's on Proteomics Informatics, it helps a lot if you know a little bit of simple programming to analyze data. That will definitely, because otherwise you're I mean, there are a lot of good tools out there that you can use, but it's still limiting if you're not able. So the after the lecture there will always be a session either on looking at a existing software package or on looking at uh, learning a little bit about Python and also uh, solving a problem that's related to the course. But in the beginning, we won't have the problems because it's only after a few weeks that we'll start with those because you'll need to learn the basics. So that's sort of and there is a website and you can go there to get updated information. And right now there is, I will add more information, a little bit about the reading material, so the slides will be up there, and if the recording succeeds, there will be the video, so the capturing the screen and the audio, and so always go there. And the other thing is so the schedule. Oh yeah, so the idea with this course is that you should be able to analyze proteomics data sets. So you should understand the basics of the different experiments, and you should be able to understand what are the different ways to approach that problem. And often there are many different ways that one can do experiments, so we're going to discuss that also. And the schedule, so the idea is that we'll meet 15 times total, and the last time is presentation of projects. So there is no exam, but there will be, you have to pick a data set, a proteomics data set, and then preferably from a publication, and then reanalyze it and go through and maybe analyze it in a different way than the authors did, or maybe try to reproduce exactly what they did, and that's and then there's a presentation at the end about 10 minutes each, and that's the examination. And then of course to do the smaller examples that we'll have after some of the classes. And any questions about this right now? How much proteomics have you guys worked with? I know several of you are from proteomics labs. So maybe we can go around and you can say how much. So you want to start?
Okay. I'm on Han, by the way. I know little about proteomics because my previous research is focusing on genomics data, so basically from the scratch.
Mm-hmm.
I'm not very knowledgeable about the proteins. Only the introduction class.
Yes. Okay.
That's much.
So, I know the theory, but I don't know how to analyze the data.
Yes. So you.
And experimental setup, I did some.
Yeah. Okay.
Yeah. So I just joined the lab. We're both in the same lab. So I know some of the theory, and I'm learning we use R as a lab to analyze our data, but I'm very new to that. So
Mm-hmm.
that's why I took this.
Okay. Mm-hmm.
I've used R before a very long time ago.
Okay, good.
It's a little primitive,
Yeah.
but yeah, that's the extent of my experience.
Okay.
I'm Heather. I've worked quite a bit with proteomics, but I've collaborated with proteomics labs, and I come from proteomics from a very different perspective. I look at it from the biology side rather than the straight informatics side.
Mm-hmm.
So I've used some softwares to analyze proteomics. I've never done my own coding, and I've also co-created a new way of analyzing proteomics for neurons because they're a little bit different than other sides of course.
Okay, great. So that's no. So it's good. So you have quite diverse background, but you know, some of you know more about programming than some of you know more about proteomics. So that's perfect. And I think we're missing one person still. So who's not coming today? Yeah. Yes. Another one.
And she's the person that I've worked with a lot. So she's also done a lot of proteomics, but not from the mass spec peptide analysis side, but from interpreting the data and figuring out what it means.
Yes. Yeah. Well, that's good. Yeah. So we're gonna here get into quite a lot of detail into the mass spectrometry. So this time I'll give a quick overview of the course. And so right today I'm going to give this overview of proteomics and start with giving you a few. I mean, you probably already are convinced that it's a good thing to work with proteomics. Otherwise, you wouldn't be here. But I'll give a few examples of. So one thing that one often can see. So this now is looking at copy number variation. So on the y-axis, you have the changes in copy number for different genes, and then on the x-axis the changes in protein abundance between. So these are two different cell types just compared, and as you see there isn't much correlation between copy number changes and proteome and changes in the proteome levels. So for example, the 20S proteasome are the ones that are marked in red, and you see that there are large changes in the copy number, but there are almost no changes in the protein levels. So this is quite common. So that's one reason why it's very important to look at the actual proteins, because there is some correlation sometimes, but often not. And this is an example of where there isn't. And people have also looked a lot at correlation with mRNA and protein levels, which should be closer, of course. And there, the correlation coefficient is around 0.4 usually in a lot of, and so there is correlation, but it's not for all genes, and it's not very strong. Another example, of course, is to look at protein complexes. So this is work by a group of people from mainly at Rockefeller, but with other collaborators that looked at the nuclear pore complex and how it was assembled, and took lots of different kinds of information. So they took ultracentrifugations to get shapes and shapes of complexes. They did affinity purification to get interaction partners, electron microscopy to get larger overall shapes. And then they put all these together and did simulations to determine roughly how this large protein complex functions. And that's something that we definitely need to work on the protein level to be able to determine how these protein complexes are put together. And of course, another thing is signaling, where as a response to different reagents, we have fast changes in the in protein levels, and especially in changes in phosphorylation of proteins that then signals to and the signal goes into the nucleus and then leads to transcriptional changes. But to understand these signaling networks, we need to know where the phosphorylation is and how it changes as a function of the stimuli. So these are a few examples. So if we then look at bioinformatics, where bioinformatics fits in biological experiments is that we have a biological system that we study, and the first place where bioinformatics comes in is to do the experimental design, and it's often forgotten that one should think about already when doing the experimental design what kind of data one and how one will then later analyze it. So that's because otherwise it's easy that it happens that one in the end doesn't have enough data. So then, of course, what most people think about bioinformatics is the data analysis. But again, think about that experimental design is also an important place to. So the basic mass spectrometry-based proteomics experiment, you start with a cell, analyze it, and then fractionate the proteins, maybe digest them typically, and then analyze them by mass spectrometry. And then it's the informatics step, how to go from the mass spectra to then identify and quantify proteins. And there we have to find the peaks, we have to determine the charge, the isotope, integrate peaks, and then do the database searching or other analysis. So this is something we're going to go through step by step how these are all done. Then the next week we're going to talk about mass spectrometry. And the basic mass spectrometer is an ion source, a mass analyzer, and a detector. And out of the detector comes the spectrum. And what we always measure is mass to charge and the intensity at these mass to charge. And then what we're going to talk about is that there are different types of ion sources, there are different types of mass analyzers, and we'll go through that. And it's and the reason why we go into this technical detail is that for when you do the experiments, it's important to know what the limitations are of different types of, when you make different choices here. And also what the simple mass spectrometer is often not used, but one can elaborate on it. And for example, this shows where you have two mass analyzer and a fragmentation chamber in between. And this is what most mass spec experiments involve. And then the idea is to use the first mass analyzer to select a specific peptide and then fragment it and then analyze its fragments. And the way most people do these experiments is that they have low energy collisions that with a gas in the fragmentation chamber, and then we get fragmentation along the peptide backbone at the peptide bond. So we get what's called either B or Y ions depending on where the charge gets retained. So if we get the charge on the N-terminal side of the fragmentation, it's called the B ion, and if it's on the C-terminal, it's a Y ion. And these are sort of the structures. So the Y ion is on its N-terminal is exactly like a peptide, so it's an NH, and on the C-terminal also it's a COOH, just like the peptide. So the only addition is the charge. And then, but the B ion has a loss of OH here. So this will get back to when we're going to calculate the mass of these fragments. So then another thing is that often we work with very complex mixtures, so we need to do a separation step, and this is often done with liquid chromatography. So what we end up doing is we have, as a function of time, we separate the peptides, often for a few hours separation, and then during this time we just collect as many mass spectra as we just can, and switching between surveying what kind of peptides are present and fragmenting them one by one to get the fragment mass spectra. So then in the third week, we're going to talk about how to analyze the mass spectra and what kind of signal processing methods we can use to be able to, and mainly to find and integrate peaks. So this just shows 3 example spectra. Here's a peptide isotope distribution, and here we have reasonably high signal to noise ratio, and then that decreases more and more, and here we can't really see the and the idea is that we want to scan this through this peptide and find where the peaks are. And we're going to go in quite detail on how to do this well. Then on the 4th lecture, we're going to talk about protein identification. And this is what most people are familiar with, is that you take fragment mass spectrometry and compare it to a database. And what we do, so in the experiment, as we said before, we take the cells, slice them, then digest the proteins and separate them with chromatography, and then collect fragment mass spectra. So in the database, in the computer, what we do then is we take a sequence database, pick one protein first, then digest it in the computer using the same rules as the enzyme that was used in the experiment. Then we pick first one peptide, and then calculate all the fragment masses, and then compare to the fragment mass spectrum. And then we repeat this for every peptide in that protein and for every protein in the database. So this ends up being a lot of calculations, but it can be done reasonably fast. And in the end, for each of these comparisons, we calculate the score, and there are many, many ways to do that. People have developed, there are probably almost up to 100 different algorithms or software on how to do that. But there are a few that are used more often, and we'll look a little bit more in detail at those. And then we rank these peptides according to how well they match the fragment mass spectrum, and then we test the significance of the match. And that means that we want to make a difference between, is this a random match or is it significant? And we will always get, there's quite a lot of randomness to this matching, because first of all we measure the mass. It's a finite accuracy of the measurement, but even if we had and nowadays we have very good accuracy though. So, but even then, different peptides, if the different masses of amino acids are quite similar in the sense that they are only composed of a few atoms. So there are only a few different masses, and if you add in that there could be modifications also, there can be a lot of randomness to it. So so the result of a search like this would be a list of proteins with some kind of measure. So here we calculated an expectation value for that this protein, in this case albumin, is in the sample. And so during these experiments we connect many spectra, so we can often identify many proteins. So we get a list of proteins and some kind of measure of how much we trust that. And we'll talk more about what an expectation value means and how we can, and different other methods to validate. So then the fifth lecture will talk about protein quantitation. And so here, if we start out with, we want to know what the concentration of a certain protein is in the sample. But what we measure is the intensity of a peptide from that protein in the sample. So we have all these steps in between where we we have losses, and these losses are different for different proteins. And then we have the digestion step here, which is the digestion efficiency can vary quite a bit depending on the local sequence around the digestion site. And of course the conversion of the peptide into a signal is very different for different peptides, and so we don't know any of most of these losses, but what we can do, if we assume that if we have a unique peptide that can only come from one protein, we still can say that the intensity of the peptide that we measure should be proportional to the concentration of the protein. And of course, this has to be tested so that we have to make sure that this is within the linear range, because at very low amounts, then we go below the detection limit and we won't be able to detect anymore. And at very high amounts, we can get saturation. But usually the assumption is that we're operating within the linear range, which then you get this proportionality. And so the one way to then use this is to what's called label-free quantitation, where we just run different samples one after the other and just try to keep everything as constant as possible. So all the conditions. And then we can assume that all these constants don't change. But of course they do change a little bit every time, but we try to minimize that. And then we can say that the ratio, if we measure a ratio of intensities, then the ratio of the proteins should be proportional, should be equal to that ratio. Then in the sixth lecture, we're going to talk about databases and data repositories and different standardization efforts that have been made. And we're mainly going to talk a lot about a database called the Global Proteome Machine Database, GPMDB. So this is a database that has been developed by Ron Beavis, and he collects public data from the public domain, and it contains a lot of different experiments. So in this is about a year old, but now it's actually almost 300,000 experiments from people all around the world that have been collected. And the idea with this is that the same as other crowdsourcing efforts is to, by having a lot of data, one can structured in a way that it's useful. People can use this to, first of all, one thing would be before doing an experiment, if you're interested in a protein, you can go in here and see what other people have seen, what they have observed from this, or one can validate results. So and what happens is that people do proteomics experiment in slightly different ways, but it's still very similar how people do experiments. So this exemplifies that. So these are just 20 experiments from GPMDB looking at one protein, and the red here shows which peptides people observe, and the green, which peptides are probably very difficult to observe because they are either too short to work well in mass spectrometry, or too long, or very hydrophobic. But as you see, the pattern is very similar. So people observe quite similar peptides, although there is some variation. So this is very good in a sense that you can, if you have an experimental observation and you can compare it and see, is it very different from everyone else's, then if you have done your experiment in a very different way, that's fine. But otherwise, if you haven't, then something is probably wrong with your experiment. And then one can do this on the mass spectrometry level. So here, for example, we are comparing one spectrum that we measured. So this is a fragment mass spectrum and of a peptide, and then we find two matches in GPMDB, and we see that the relative intensities are quite similar. And that's one thing that we see in if the same fragmentation technique is used, then the relative intensities of the peaks in the fragment mass spectrum depend on the sequence of the. So that's sort of a signature that we can use to validate also. So then the seventh lecture is about de novo sequencing. So here now we have a spectrum, but we don't have a database to search. So we have to just, without the database, determine what is the sequence. So what we have at our hand is, of course, the masses of the amino acids and of potential modifications. And then we look at the mass differences in these spectra and try to match it with the different amino acids. And then come up with sequences that are consistent with the spectrum, and preferably all the sequences that. And why? Preferably we want one sequence, but that doesn't. Often we have missing information, so we don't have fragmentation between each pair of amino acids, and then we won't be able to get the full sequence completely determined. But this is something that you'll try to, you can work with a little bit. But I mean, often people try to avoid this because it's much more time consuming than database searching, but sometimes it's unavoidable. Then one can do the experiment in several different ways, but there are two main sort of ways to design proteomics experiment. Either you can do what's called the shotgun experiment, or a discovery experiment, where you don't make any assumptions, but you go in and you try to fragment as much as possible and get as much information as possible to get a survey of what's in your sample. Then, as opposed to that, we have the targeted, where you have a list of proteins that you're interested in, and you take select a few peptides for each, and just collect information on these peptides from these few proteins. So these are the two. And what you can do, you often, when you do the discovery proteomics, you can identify a lot of proteins, and you can also, depending on how you quantify a lot of proteins, but you get much better quantitation when you do the targeted analysis. So then the ninth lecture, we're going to look at how one can combine genomics and proteomics. So this example shows it for how we can make a tumor-specific protein database. So usually when we search, we just download a reference database from somewhere. So in this case, from we used Ensemble, and that we can identify often quite a lot of the proteins. But in some cases, what we can do is, for example, for tumors, there are lots of genomic rearrangements. So first we take the germline sample, for example, from blood, and sequence the genome to get the germline variants of this individual. And then we can do the same for the tumor sample to get the somatic variants. And if we do sequence the RNA, we can look for alternative splicing events and expression in regions that are not annotated in the database, and add that into to make a sort of tumor-specific database. And so the things that we expect to see is, for example, known exons. Let's say these 3 exons are known for this gene, but in the database we only have annotated that exon 1 is connected to 2 is connected to 3. But in this case, we might in the RNA-seq see that exon 1 is connected directly to exon 3. We can find exons that are not annotated in the database, fusion genes, and again, single nucleotide variants or small insertions and deletions. Then we're going to talk about post-translational modifications, and that's a lot of people are very interested in it, especially when working with signaling. So this example just shows that on top of the identification here, we also need to localize exactly where the modification is. So in this case, what we have is the modification can either be here in the blue location or in the red location here. Then it only counts to distinguish between these two the fragments that we have in between these two. So in this case, what we have a matching spectrum. So we have, in this case, much more evidence for that the localization here, the blue, but there is a little small peak here that's inconsistent with the blue and instead supports the red. So then the question is, which assignments should we say that in this case that it's only in position 1, or should we say that it's in a mixture of both, or that it's we can't really tell? So in this case, probably what in most cases what one would do is to say that it's 1, but there are more. There are cases that are less clear than that. Okay, so not every. I mean, even though we do mostly cover mass spectrometry, there is a lot of proteomics that's done with other methods. For example, looking at protein-protein interaction with microscopy. So we're going to have one lecture on extending the signal processing to 2-dimension on how to analyze images and looking at protein-protein interactions. This shows a few cardiac cells. So here is one cell there, and it's these long cells, and then another cell up here. So these are the junctions between the cells. And in this case, we looked at plakophilin 2 and connexin 43 interactions using super microscopy. Then we're going to talk about more in detail about protein-protein interactions in lecture 2. So both by mass spectrometry and by microscopy. So this is an example. So if you're interested in a protein complex that contains a few different components, we often tag one of the components, the known component, and then this is of course in a complex mixture. So we have a lot of other proteins in the cell, and then we often use a magnetic bead to pull out the tagged protein, and the idea is to try to not disrupt the wash away as many of the irrelevant proteins as possible, but not disrupt the actual complex. But what happens in reality is that we always have a few contaminants left, and we lose part of the complex. So this is, and then in the end we analyze this by mass spectrometry, but what we'll talk a lot about is how can we tell which is a contaminant, and how can we can we do the experiment so that as many of the real proteins that are really part of the complex, how we can preserve that. So then week 13 we're going to look at how to analyze and visualize data, and we're going to do that throughout the talk, but there will be one lecture more dedicated to how we can in a good way analyze and visualize data. So this for example just shows two tumors, one basal breast tumor and one luminal breast tumor, and so we've done an experiment and we've just done spectrum counting, a simple way to do the quantitation, and we're just comparing the two, the number of the intensity, I mean the number of counts we get for the basal for each protein and the luminal, and we see that it's quite correlated, but there are outliers, and then there are lots of proteins that we just observe either only in the luminal or only in basal. And of course we can show this in another way, where we instead take the log ratio here on the x-axis, so we sort of turn the, and this one is probably much easier to look at and determine which ones are the outliers that are very different between the two. So then in week 14 we're going to see how this can be used to in diagnostics, to make use the proteomics results for molecular signatures, and we'll also go through a little bit the history of proteomics in the clinic or in translational research. So molecular signature, what we mean by that is a panel that contains either many genes or many proteins that can distinguish from different disease states. So in this case it's just two types of leukemia, and you can see that these gene panel distinguishes very clearly between the two types. And so what we can use these signatures for is, for example, we take a biopsy and then take a profile, in this case a gene profile, and compare it to a molecular signature to decide whether it's primary or metastatic lung cancer in this case. And usually we don't just want to make these distinctions, but we want to then use those to change the treatment and improve the way so that everyone can be treated the right way. Because often there are for a certain tumor there are many choices of treatment, and it's not always clear, and the doctors it's not unusual they pick one chemotherapy at random. Now, but it would be much better if one could choose, have a signature that could tell which treatment should be used. So then the last time we meet, then it's the presentation of the projects. And you can pick any data set you want. You can generate it yourself, or you can pick it from the literature. It's not, yeah, it's up to you. And one place where there are highlighted data sets is this address, the gpm.org. And that's, if you don't know where to start, that's a good start to. And so Ron Beavis there has written short introductions and short overviews what the data sets are about. And then 10-minute presentations so this will be sometime in May. And oh yeah, so this is how it looks, that the data sets are annotated, and there's a short description of them, and there's a link to the paper, and also how to download the data, usually from different public sites. So this is the lecture for today. So if you need to contact me, email me, and look always on the website for the course. And if you have any questions now.